2025 to 2026 academic year is cellular dysfunction, the root of human disease. Over the next term, we will be exploring the mechanisms and manifestations across the lifespan as part of this advanced pathophysiology, graduate-level course. By the end of this module on cellular dysfunction, you should be able to meet 4 key learning objectives. First, you must be able to analyze fundamental mechanisms. This includes examining oxidative stress, protein misfolding, DNA damage, and epigenetic alterations at the cellular level. Second, we'll correlate with disease etiology. We will connect these specific cellular dysfunctions to major human diseases, including neurodegeneration, cancer, and metabolic syndrome. Third, we will examine across the lifespan. This involves analyzing disease manifestations from a cellular perspective across different age-related conditions. Finally, you will need to evaluate interconnected pathways. We will assess how these cellular pathways interact and often exacerbate disease progression. The central premise of this course is the cellular foundation. Most human diseases, from neurodegenerative disorders to cancers, fundamentally originate from disruptions at the cellular level. A healthy cell maintains intricate homeostatic mechanisms, but when these are compromised, dysfunction ensues, leading to the pathological states we recognize as disease. This dysfunction manifests across a wide range of major human diseases. Neurodegenerative disorders, such as Alzheimer's, Parkinson's, and Huntington's. Carcinogenesis, covering various cancers that have cellular origins. Metabolic syndrome, which includes conditions like diabetes, obesity, and hypertension. And finally, other pathologies driven by things like inflammation, senescence, and mitochondrial dysfunction. To truly understand disease etiology, we must examine the fundamental molecular mechanisms that disrupt normal cellular function. This overview highlights the 3 key interconnected mechanisms that we'll be focusing on. First, oxidative stress. This is an imbalance between the production of reactive oxygen species and the cell's ability to detoxify them, leading to cellular damage. Second, protein misfolding. A disruption of protein homeostasis results in protein aggregation and cellular toxicity. And third, DNA damage and epigenetics. These are genetic alterations and epigenetic modifications that underpin disease development. The key concept here is that these mechanisms interact and exacerbate each other, creating complex disease etiologies. Disease manifestations are the result of disruptions in these interconnected mechanisms. To truly understand disease etiology, we must examine the fundamental molecular mechanisms that disrupt normal cellular function. This overview highlights the 3 key interconnected mechanisms that we'll be focusing on. First, oxidative stress. This is an imbalance between the production of reactive oxygen species and the cell's ability to detoxify them, leading to cellular damage. Second, protein misfolding. A disruption of protein homeostasis results in protein aggregation and cellular toxicity. And third, DNA damage and epigenetics. These are genetic alterations and epigenetic modifications that underpin disease development. The key concept here is that these mechanisms interact and exacerbate each other, creating complex disease etiologies. Disease manifestations are the result of disruptions in these interconnected mechanisms. Let's break down oxidative stress. At its core, it is an imbalance between the production of reactive oxygen species, ROS, and the body's ability to either detoxify them or repair the resulting damage. The core components involve ROS itself, the cell's antioxidant defense, such as SOD, CAT, and GPX, and the resulting oxidative damage to cellular components. The common types of reactive oxygen species we deal with include superoxide radicals or O2, hydrogen peroxide, and the highly destructive hydroxyl radicals, OH. This damage is not selective, it affects all cellular components, leading to lipid peroxidation in the cell membrane, protein modifications like carbonylation and nitration, and base modifications or strand breaks in DNA. Protein misfolding refers to the disruption of normal protein folding processes. This leads to the accumulation of misfolded proteins, triggering a cellular stress response that can ultimately result in cell death. A key event is ER stress, which occurs when the endoplasmic reticulum's capacity to fold proteins is overwhelmed. This initiates the unfolded protein response, UPR, a stress response that attempts to restore homeostasis by increasing chaperone production and reducing overall protein synthesis. However, when the UPR fails, it leads to chronic ER stress, which triggers apoptotic pathways. The accumulation of these misfolded proteins has profound pathological significance. They can aggregate, forming insoluble deposits characteristic of neurodegenerative diseases. A classic example is the formation of amyloid plaques in Alzheimer's disease. The key takeaway is that protein misfolding represents a critical point for intervention in disease progression. Moving on to DNA damage and epigenetics. DNA is constantly exposed to damaging agents from both endogenous sources like ROS and replication errors, and exogenous sources like UV radiation and environmental toxins. While cells have repair mechanisms, failures allow mutations to persist and accumulate, leading to genomic instability, which is a hallmark of both aging and cancer. Epigenetics refers to heritable changes in gene expression without any change to the underlying DNA sequence. The key epigenetic modifications include DNA methylation, histone modifications, and non-coding RNA regulation. These aberrant epigenetic changes are highly problematic in disease because they can silence tumor suppressor genes or activate oncogenes. Critically, DNA damage and epigenetic alterations work together, collectively contributing to the hallmarks of cancer. We'll use Alzheimer's disease as our model for progressive neurodegeneration. This disease is characterized by progressive neuronal loss and cognitive decline, which is a perfect example of how cellular dysfunction drives pathology in the brain. The disease is defined by two pathological hallmarks: amyloid-β plaques. These are extracellular accumulations of misfolded A*/β peptides. These insoluble plaques initiate a cascade of synaptic dysfunction and inflammation. Tau neurofibrillary tangles. Here, hyperphosphorylated tau protein aggregates into intracellular tangles. Normally, tau stabilizes microtubules, but in AD, it detaches and forms these aggregates, severely disrupting axonal transport and neuronal integrity. The underlying mechanism connecting these is oxidative stress, often exacerbated by mitochondrial dysfunction. Oxidative stress accelerates both A*/β aggregation and tau hyperphosphorylation, creating a devastating feedback loop that further damages neurons. The final result is a progressive network disruption that spreads throughout the brain. Carcinogenesis, the development of cancer, is the result of accumulated DNA damage and epigenetic alterations that disrupt cellular growth control mechanisms. The DNA damage arises from endogenous sources like ROS or exogenous mutagens. This damage can lead to mutations in critical genes, specifically proto-oncogenes and tumor suppressor genes. Simultaneously, epigenetic alterations such as DNA methylation and histone modifications can silence tumor suppressor genes or activate oncogenes without changing the underlying genetic sequence. Together, these mechanisms lead to the classic hallmarks of cancer, including sustained proliferative signaling, evasion of growth suppressors, resistance to cell death, and the activation of invasion and metastasis. Metabolic syndrome is a cluster of conditions, including insulin resistance, obesity, hypertension, and dyslipidemia, with type 2 diabetes mellitus being the most prominent manifestation. The cellular dysfunction here involves several key processes: mitochondrial dysfunction, characterized by impaired oxidative phosphorylation. This dysfunction leads to lipid accumulation. Lipid intermediates, such as diacylglycerols and ceramides, interfere directly with insulin signaling pathways. Oxidative stress is increased by chronic hyperglycemia and mitochondrial dysfunction, which damages cellular components and impairs insulin action. The final result is insulin signaling pathway interference. Lipid intermediates activate serine kinases that phosphorylate insulin receptor substrate proteins, inhibiting the downstream signaling cascade. Cellular senescence refers to cells that have permanently escaped the normal cell cycle checkpoints and entered a state of permanent growth arrest. These cells, though not dividing, are highly active and contribute to chronic inflammation by releasing inflammatory cytokines like IL-6, IL-8, and TNF-alpha. They also contribute to tissue dysfunction by releasing matrix metalloproteinases that degrade the extracellular matrix. Senescence impacts nearly all major affected organ systems, including the brain, heart, kidney, lung, and blood vessels. It is directly associated with age-related diseases such as atherosclerosis, osteoporosis, and Alzheimer's disease. What is mitochondrial dysfunction? It is a disruption in the energy-generating capacity of our mitochondria, resulting in two critical outcomes: reduced ATP production and increased reactive oxygen species, ROS, generation. The reduced oxidative phosphorylation leads to decreased ATP synthesis, severely affecting cellular viability and function. The excess ROS production contributes directly to cellular damage and disease pathogenesis. This single mechanism is associated with a staggering list of diseases: neurodegeneration, Alzheimer's and Parkinson's, cardiovascular disease, heart failure and arrhythmias, diabetes mellitus type 2 and insulin resistance, respiratory disease, COPD, hematological disorders, and, as we mentioned, metabolic syndrome. Inflammation dysregulation is characterized by the chronic, persistent activation of inflammatory pathways without an appropriate resolution, leading to sustained tissue damage. This is implicated in nearly every disease we've discussed, including neurodegeneration, carcinogenesis, metabolic syndrome, and others like atherosclerosis and inflammatory bowel disease. The key mechanisms involve a chronic release of inflammatory cytokines, an impairment of anti-inflammatory responses, and dysregulated cellular turnover. This creates a vicious cycle where cellular stress leads to an inflammatory response, which leads to persistent activation, tissue damage, and further cellular dysfunction. The mechanisms of cellular dysfunction manifest differently across the lifespan. In developmental disorders, we see mitochondrial dysfunction, oxidative stress, and DNA damage. As we move into early adulthood, protein misfolding, oxidative stress, and inflammation become more prominent. Middle age is characterized by the accumulation of cellular senescence, mitochondrial dysfunction, and DNA damage. In late adulthood, we see the overt presentation of neurodegeneration, metabolic syndrome, and epigenetic alterations. Finally, the major geriatric conditions are Alzheimer's disease, cancer, and metabolic syndrome. The key takeaways are that cellular senescence accumulates throughout life, and mitochondrial dysfunction is a major contributor to aging. Crucially, early interventions targeting these mechanisms may significantly modify the disease progression later in life. This understanding leads us to powerful therapeutic implications. We are seeing a paradigm shift in disease treatment, moving from merely treating symptoms to targeting root causes at the cellular level. This includes strategies like senolytics, which specifically target and clear out senescent cells, antioxidant therapies to mitigate oxidative damage, epigenetic drugs to correct aberrant gene expression patterns, and metabolic interventions to target mitochondrial dysfunction and lipid intermediates in insulin resistance. These approaches represent a fundamental and promising shift in how we prevent and treat chronic disease. Finally, here are the references detailing the key scientific literature that informs this presentation, covering both the mechanisms of dysfunction and the disease manifestations we have discussed today. Please review these resources for a deeper understanding of the material.